it me, talking to you extraordinaire. Still riding that DNA momentum with an episode about the fascinating event we call transcription, the first step of gene expression. All will be revealed in time. And if there's one thing people love more than triumph, it's annoying vagueness. Anyways, on to my cool words I had to say. If you watched the previous video covering DNA replication, you may notice this process initiates a very similar manner, but the respective purposes are very different. The end goal with transcription being the processing of DNA sequences to produce RNA. The protagonists of this style fest are enzymes called RNA polymerases, of which there are 3 main types, each responsible for transcribing 3 different types of RNA. RNA polymerase is a bit of a handful. From here on, we're going to be shortening it to RNAP. It's simplified, and any excuse to say RNAP I will gladly grab. Speaking of things that come in sets of 3, transcription itself has 3 primary steps: initiation, elongation, and termination. And golly gee willers, doesn't that sound a wee bit familiar? The big 3. Initiation. RNAP cruises along the DNA strands calmly until it recognizes a promoter sequence, whose job is exactly what it sounds like, and immediately anchors onto it. Much like helicase in the previous video, RNAP begins to unwind the double helix and uncouple the bonded base pairs. RNAP will now solely use one side of the DNA molecule to build its RNA with. This will be known as the template strand. Floating alongside RNAP are molecules known as ribonucleotides. They act as building blocks that RNAP uses to build its own sequences with, becoming base pairs for the mRNA sequence to be. DNA uses the base pairs AT and GC, but RNA uses U instead of a T, U representing the molecule uracil. Uracil is structurally pretty dang similar to thymine, so why replace it at all, you might ask? Well, uracil is essentially a more resourcefully efficient molecule to use. DNA molecules stick with thymine though, as it makes unwanted damage or changes to DNA easier to detect. Initiation. RNAP chugs along the template strand, gathering ribonucleotides, and binds them covalently with phosphodiester bonds. After reading several base pairs and stringing together the proper ribonucleotides, RNAP gradually plumps out a sequence of code that we can call Chauncey. I mean mRNA. mRNA, like DNA, is built in that good old-fashioned 5' to 3' direction. At least it's keeping up consistency for those of us wanting to memorize this material. Then the last phase of termination. This step is fairly self-explanatory. RNAP runs into a stop sequence which, and you better write this complicated portion down, stops transcription. Once RNAP finds this sequence of code, it releases its template strand and goes back to floatin' and boatin' until it hits the next promoter sequence. Well, there you have it, lads. Be sure to subscribe. Maybe share a share. Post a link to your friends, possibly cram a jamming for an upcoming exam.